Today we are going to continue talking about development of embryo. We have already finished the first week and second week in our previous videos. If you guys haven't seen the videos, just go and get the link in the description of this video. We have talked about the development up to the second week. In first week we have covered fertilization, cleavage and blastocyst formation. While in the second week we have discussed about the implantation of blastocyst, that how blastocyst is buried into endometrium of uterus, and as a result what testicular reactions occurred, and then we went into all the major events of second week. Guys, so far we have learned that some major changes have been occurred in the epiblast and hypoblast, and as a result of those changes some important structures have been formed during the second week. For example, formation of bilateral embryonic disc, formation of amniotic and exocelemic cavities, establishment of uteroblasm to circulation and primary chorionic villi. Now let's discuss about further developmental changes that are going to take place in third week. And before we move towards third week, first let me remind you guys to subscribe the channel and press the bell icon for notification of our next videos. So major events of third week are gastrulation, formation of Notochord, formation of Intraembryonic coelom, development of somites, formation of primitive cardiovascular system and chorionic villi. Let's begin with gastrulation. It's one of the most important and significant event of third week. In the gastrulation, 3 germ layers are formed: ectoderm, mesoderm and endoderm. As you guys know, in the second week, after differentiation of embryoblasts, we got 2 layers, epiblast and hypoblast, and these 2 layers formed bilaminar embryonic disc. Now on that embryonic disc, a new structure is going to be formed, that is primitive streak. So gastrulation begins with the appearance of primitive streak on the dorsal surface of embryonic disc on day 15. The primitive streak can be seen in the longitudinal line of the coiled part of embryonic disc. The groove within the primitive streak is called primitive groove. By the 60th day, a small circular swelling appears at the cranial end of the primitive streak. This swelling is called primitive node, and within the primitive node, a small depression appears that is called primitive pit. As the primitive streak is formed on the dorsal surface of the embryonic disc, so you guys can understand that epiblast and hypoblastic cells must be lying somewhere around the primitive streak. So next changes are going to appear due to the migration and displacement of epiblastic and hypoblastic cells. And these epiblastic cells actually invaginate the primitive streak and primitive node at different positions. Some caudally, some cranially, and others through midline. Let's discuss about the formation of 3 germ layers. So first we will discuss about the formation of embryonic endoderm. Some of those epiblastic cells is invaginates through the middle part of primitive streak, invades and displaces the cell of hypoblast. Eventually, the hypoblast is completely replaced by a layer of roughly cuboidal cells. This layer is called embryonic endoderm or definitive endoderm. Next is embryonic mesoderm. Those epiblastic cells which migrate through the coiled part of primitive node and major part of the primitive streak give rise to a layer of cells between the epiblast and newly formed endoderm. This layer is called embryonic mesoderm or intraembryonic mesoderm. Guys, the last one is embryonic ectoderm. Although most of the cells of epiblast migrates through primitive node and primitive streak, a larger number of epiblastic cells do not invaginate and give rise to a new layer of columnar cells on the dorsal surface of embryonic disc. This layer is called embryonic ectoderm or simply ectoderm. So guys, till now we have got 3 layers in the process of gastrulation: ectoderm, endoderm and mesoderm. Mesoderm is formed between the ectoderm and endoderm. Now this mesoderm will further be organized into 3 segments: paraxial mesoderm, intermediate mesoderm and lateral plate mesoderm. So guys, as a result of gastrulation, the bilaminar embryonic disc consisting of epiblast and hypoblast becomes converted into 3-layered structure called trilaminar geminal disc. All tissues and organs of the human body are derived from these 3 layers. Guys, as we know, major changes in the third week are taking place due to the migration of epiblastic cells towards the primitive streak and primitive node. So the earliest epiblastic cells that invaginate through the cranial most part of the primitive node form a compact mass of cells called precoidal plate. Here I'll mention some other structures which are also appearing in third week of development. So these structures are oropharyngeal membrane, cloacal membrane, and cardiogenic plate. First we will see how oropharyngeal membrane is formed. There are some regions where ectoderm and endoderm remains adherent to each other. One of these regions is called oropharyngeal membrane, and it is located near the cranial end of embryonic disc, whereas the other one is situated near the caudal end of the disc and is called cloacal membrane. Now we will see how cardiogenic plate is formed. Guys, those mesodermal cells which passes cranial to precaudal plate and oropharyngeal membrane form a cardiogenic plate, which is the site where heart will develop. Now let's discuss about the formation of notochord. So guys, in the formation of notochord, first notochordal process is formed. So guys, those plastic cells which invaginates into the cranial part of primitive pit are known as pre-notochordal cells. Now these cells give rise to tubular structure called notochordal process. Next is formation of notochord. By the 80th day, the floor of the notochordal process fuses with the underlying endoderm. The fused layers soon degenerate and disappear, resulting in the loss of floor of the notochordal process. After the disappearance of the floor, the roof of the notochordal process remains in the form of curved plate, which is called notochordal plate. Later, the cells of notochordal plate detach from endodermal cells, proliferates and gradually roll up to form definitive notochord. Next, during the third week is formation of intraembryonic coelom. On the 80th day, a number of small spaces appear in the lateral mesoderm of both halves of the embryonic disc and in the mesoderm of cardiogenic plate. These spaces soon coalesce to form a U-shaped space, which is called intraembryonic coelom or intraembryonic cavity. Guys, the formation of neural plate is also taking place in third week. The precaudal plate and notochord induce the overlying ectoderm to form a elongated patch of thickened epithelial cells called neural plate. Next is formation of somites. We know that intraembryonic mesoderm has three parts: paraxial, intermediate, and lateral plate mesoderm. Now we will see how segmentation of paraxial mesoderm is playing its role in the formation of somites. The paraxial mesoderm further segmented to form a series of cuboidal cells called somites. This segmentation of paraxial mesoderm occurs in gradual and progressive manner. It begins in the cranial region and proceeds in the caudal direction. Now these paired segmentation of paraxial mesoderm appears as a circular disc-like structures called somatomers. Now all the somatomers paired give rise to cuboidal masses of epithelial cells called somites. Now let's talk about the formation of primitive cardiovascular system. Guys, during the first week of development, the conceptus obtains nourishment by absorbing nutrients from the secretion present in the lumen of the uterine tube and uterus. In the early part of second week, the syncytial trophoblast erodes the endometrium and breaks down the decidual cells of endometrial stroma. As the embryo begins to grow rapidly in the third week, it is no longer able to satisfy its nutritional requirements simply by diffusion. To obtain nourishment and oxygen directly from the maternal circulation, a primitive cardiovascular system develops and begins to function by the end of the third week of development. With the onset of third week, at 50th day, the blood vessels begins to form in the extraembryonic mesoderm of yolk sac, connecting stalk, and chorion. Intraembryonic blood vessels begin to develop by the 70th day of development. The basic process of blood vessel formation is called vasculogenesis. So guys, in the process of vasculogenesis, we get a network of endothelial tubes called primitive capillaries, and the heart develops in the cardiogenic plate. So cardiovascular system is the first organ system to reach functional state. By the 20th day of development, the primitive heart links up with the primitive blood vessels, and the primitive heart starts to beat by the end of third week. So the circulation of blood through the primitive cardiovascular system begins. Let's discuss further development of chorionic villi. As we have already discussed the formation of primary chorionic villi in the second week of development. So here we will discuss further development of chorionic villi. Guys, early in the third week, mesenchymes grows into primary villi, forming a core of mesenchymal tissues, and forms secondary chorionic villi. Now some mesenchymal cells in the secondary chorionic villi will differentiate into capillaries and blood cells, forming the tertiary chorionic villi. In the last, we will talk about allantois. On about 60th day, a narrow tubular diverticulum grows out from posterior superior wall of the yolk sac and extends into the extraembryonic mesoderm of connecting stalk. Guys, this blind diverticulum is called allantois. This is also called allantoenteric diverticulum. Guys, third week ends here. If you guys have any questions regarding this topic, you can ask in comment section. And if you like the video, share it and subscribe the channel. Thank you.